Welcome back to part 2 of our screencast for the second module in this unit. From here we're going to start talking about actual different types of cell division and how they occur. So in part 1 we discussed how DNA actually gets replicated in cells and it has to happen before cell division. Now we also talked about there being two different types of cells. You have somatic cells and sex cells, aka gametes. Now, because we have two different types of cells, we actually have two different ways of dividing them. And it depends on what kind of cell you're trying to create. So if you start with a somatic cell and you are trying to get more somatic cells, then you're going to divide your cells through the process called mitosis. Okay. Now, I'm really going to try and emphasize names here because the two types of cell division have very similar names. So from somatic cell to more somatic cells, it is mitosis. You can think of it this way: mitosis is what happens in my toes, and my toes are just normal body cells, the normal cells part of my body, or normal somatic cells. So that's one way of thinking about it. Now, if you, however, want to create sex cells, or you want those gametes, so either sperm or egg cells, then you have to go through a different process. And it's a bit longer, it's a bit more complicated, because you have to produce something a bit different to what you started with. And so this process is meiosis. Okay, so mitosis for making more somatic cells and meiosis for making more gametes, or for making gametes. And so very similar sounding processes have a lot of similarities in how they go about occurring, but they are for different purposes. So mitosis to make somatic cells and meiosis to make gametes. Those are our two different processes, and we're going to start by looking at mitosis in a bit more detail. So with mitosis, you're going to start with one cell and you're going to make two daughter cells. Any cell you start with is called a parent cell. Any cell you end up with is called a daughter cell. No particular reason other than that's the standard naming convention that we use in science. So mitosis starts with one parent cell and it will make two daughter cells. Now the two daughter cells are genetically identical to the parent cells and to each other. Now, one thing to take note of, and I mentioned this in the part 1, is this little box over here that's talking about what diploid means. So just to reiterate with that, the word diploid is referring to a cell where you have two sets of chromosomes. So it means there's two of each type of chromosome. So you're able to pair them up. So for example, you'd have two chromosome number 4s, two chromosome number 17s. That's what the word diploid means. So if N is just sort of one set of chromosomes, then 2N is saying there's two sets of chromosomes, and that's what diploid is. All right, so you can see here we start with a parent cell that is 2N, it's diploid. Now it does have to undergo DNA replication, and once it's undergone DNA replication, technically, because it's copied all its DNA, it technically at this point has double what a diploid cell would have. So instead of having 2N, it actually has 4N. But because of how DNA replication works and the fact that the copies are still attached to each other, you don't actually change the number of overall chromosomes. But technically you now have double the amount of genetic information. Then we've got all this genetic information, and then it goes through the whole process of mitosis and it divides into two new cells. And if you take 4 and divide that by 2, you end up with 2. So you can end up with 4N, dividing that into 2, you end up with two sets of 2N cells. So that's what this image here is showing. So a sort of simplified overview of what's actually happening in mitosis, we can sort of break it down to 3 key steps. So you've copied the DNA in the somatic cell, and now it's going to go through mitosis and divide into two new cells. So first of all, you have all your duplicated chromosomes line up in single file along the equator, along the middle of the cell. Then the copies get pulled apart. So if you remember, our DNA normally is like these single so chromosomes here where they're just like the letter I. Then when it copies, it becomes like the letter X because you've got a copy attached to a copy at the middle bit. So this was called the centromere. And so they're attached, the two different copies, the two new copies of DNA are attached to each other at this point. And so if we line them up single file, we can pull one copy one way and one the other way. So we can rip apart those two new copies. It's a little bit like putting a bit of paper or something through a photocopier and coming out stapled on the other side. So our two new copies are stapled together, and then it goes through mitosis, and part of that is ripping those two copies apart. So the copies of the chromosome so duplicated chromosomes line single file along the equator. The copies get ripped apart with one copy from each of those duplicated chromosomes going to the opposite end. And then once we've got sort of half of the copies on one side and the other half on the other side, the cell will pinch inwards and it will make two new cells. And so that's sort of a simplified overview of mitosis. Okay, to go through the whole process of mitosis in a little bit more detail and also sort of to animate it and hopefully make the process a bit clearer, we're going to watch a video.
Ever wish you could clone yourself? Ever wondered how you grow? In this video, you are going to learn exactly how our bodies do this. Well, maybe not quite clone yourself exactly, but your cells do it all the time. There are two types of cell division, mitosis and meiosis. Mitosis produces identical copies of cells, and is how most of our cells divide. And then there is meiosis for our gametes, because they are different. Remember that gametes only have 23 chromosomes, rather than the normal 23 pairs. In this video we are going to learn all about mitosis, the process of cell cloning. But before we start, to make sure you're not in for a rocky ride, you need to know what all of these terms mean. Pause the video and have a look. Right, so back to mitosis. Mitosis produces identical copies of cells and is involved in growth, cell repair, and asexual reproduction, which is how simple organisms reproduce. When cells divide by mitosis, the number of cells increases and hence the organism grows. So let's get stuck in. All cells contain two sets of chromosomes. So in humans, we have two sets of 23 chromosomes, making 46 in total. But to keep this simple, we are just going to look at what happens to one of these pairs of chromosomes. Drawing out 23 pairs each time would make it very busy. These chromosomes are homologous, which means they carry the same genes, but they may have different alleles, which are alternatives of the same genes. So the chromosome from your dad may carry the blue eye allele, and the one from your mum may carry the green eye allele. The DNA is exactly replicated, and each chromosome now becomes two identical chromatids, joined by the centromere. Chromatid is the word we use to describe the newly copied chromosome, the identical copies. The chromosomes become attached to spindle fibers, which run from one pole to another. The individual chromosomes line up at the equator of the cell. The spindle fibers shorten and the centromere divides, so that each chromosome becomes two separate chromatids. The cell membrane pinches to separate the two sets of chromatids into two cells. So the original cell has become two daughter cells. The daughter cells are identical to one another, identical to the parent cell, and have the same number of chromosomes as the parent cell. There is of course much more detail than this, but this is all we need to know for now. If you want to learn more, just Google mitosis. Okay, so maybe you can't go out and clone yourself, but you do at least know how your cells manage to do it. From this video, you should now know that mitosis is how cells divide in order to grow and repair, or for asexual reproduction. When a cell divides by mitosis, two new cells form, and each cell is identical to the other, also to the parent cell.
Okay, so we've talked about mitosis, which is where we are taking a somatic cell and we're basically making a brand new copy or brand new two copies of that somatic cell from it. So somatic cells always give rise to two new identical somatic cells through mitosis. But now we want to stop and look at meiosis. And meiosis is a very similar but also very different process. So the whole point of meiosis is we are making gametes. Now, meiosis is going to occur in the testes in guys and the ovaries in girls. So those are the tissues that this is actually going to happen in. It won't happen anywhere else in the body, or at least it definitely shouldn't be happening anywhere else in the body. If it is, you've got a problem, go see a doctor. All right, now what is happening here is a special type of cell known as a germ cell, and these are the only cells that actually go through this process to give rise to gametes. So germ cells themselves start off as normal diploid cells, very much like all the somatic cells in your body. But they go through a different process, and this is the process of meiosis, and they're going to produce haploid gametes. Now, I mentioned the word haploid when we did part 1, but if you've forgotten, it's down here, so you can read over that. Just to summarize, it's where we're talking about a cell that has one set of chromosomes. So there's only one of each type. So you've only got one chromosome number 4 and one chromosome number 17 or something like that. Okay, whereas diploid, which is what our somatic cells are, have 2 of each kind. Okay, now we're taking a diploid cell, those are our germ cells, and we are producing haploid gametes. So unlike in mitosis, where we take diploid cells and we make new diploid cells, we're actually not doing that. We really are halving the number of chromosomes that we have here. Now, to do this, meiosis actually is a much longer process than mitosis. So we actually end up going through 2 stages, and we end up producing 4 daughter cells instead of 2, like in mitosis. And the reason for this is a lot of the starting part of meiosis is really similar to mitosis. You replicate the DNA, you make a new copy of it. And so you end up going from a 2N parent cell to a 4N cell before it goes through this division. And so for, meiosis to actually end up with cells that are just N or haploid at the end, if you have 4N in your starting cell, you end up having to produce 4 single cells to do that. So this is why it's a longer process, and we have to go through 2 stages to get those 4 daughter cells. Now, those daughter cells, or your sperm and egg cells, they are only going to have 23 chromosomes, and that's because they're haploid and they only have 1 of each type. There are no pairs of chromosomes here. Okay, and that also means they're only going to have 1 sex chromosome. Now, meiosis is actually really important. You might like, why do we do this? Why bother? It's really important because it's actually giving, variation to a species. There are a few steps within meiosis that help vary the genetic material up a bit more, and also the fact that when you make a baby using gametes, you end up getting gametes from 2 genetically different organisms, and so that creates a baby that is genetically different to both its parents, and that's going to add variation to the whole process as well. If you've been watching, you might have noticed this little animation down the bottom. It's actually playing through the process of meiosis. I'll wait a moment for it to get back to start. Here are our first part. We've got our chromosomes actually are in pairs, and the pairs all line up and the pairs get separated. So unlike in mitosis, where you actually rip apart the copies, we're actually separating the pairs to begin with. And we make 2 new cells, and then we go through a process that's really similar to mitosis, where we separate the copies, and then we divide into, those 2 cells divide into 2 more cells themselves. And that's how we end up with 4. So we do one stage where we divide into 2 cells, and then another stage where those 2 cells divide into 2 cells themselves. And so it goes 1 divides into 2, and then those 2 divide into 2 each, so it ends up with 4. So here's kind of a bit more of an overview of what's happening in meiosis. And like I said, you've got 2 divisions. And with that first division, your pairs of chromosomes separate. So you can look either in this bottom diagram is a bit more detailed, or you can look at this more simple one here. But I'll just point to the more simple one for a moment. So we've got our first cell here is a cell where the chromosomes haven't undergone DNA replication. And that's why the chromosomes look like the letter I. Then they go through DNA replication and the 2 copies are attached to each other. So now they kind of look like X's because you've got 2 copies attached at the middle point there. Think of that middle point, that centromere, like a staple or something. What happens here is this cell then goes through the first division in meiosis. And so these 2 chromosomes are a pair. And the first division in meiosis, those pairs are separated. So the copies don't get ripped apart. That's like what happens in mitosis. That's not happening at this point. So the yellow copy goes over here and the blue one goes over here. Sorry, the yellow chromosome goes here and the blue one goes here. Then it goes through its second division. And the second division is like mitosis. And that's where the copies get pulled apart. So these 2 chromosomes, they're copies of each other, they're attached at this point. They get ripped apart. So one ends up in this cell and one ends up in this cell. And the same for the blue ones. So the second division is where your copied chromosomes are being separated, very, very much like mitosis. All right, you can see that down here in this diagram as well. So we have a parent cell, the chromosomes replicate, they look like X's at this point. They line up at what's called the equatorial plate. Don't worry about that, it just means the equator of the middle bit. Some chromosomes may swap bits, be a bit fancy like that. It's called crossing over. Again, not something to worry about. It's just the way they sort of introduce extra variation. But basically they line up and then the pairs of chromosomes separate. So you can see these pairs go this way and the bluish pairs go that way. Then our cells basically divides and we end up with essentially 2 new cells here. Then we go through the second division, and like I said, the second division much more like mitosis. Our chromosomes line up single file, and the copies get pulled apart. So you can see they get ripped going. So you've got, this is 2 copies, 1 copy goes that way and 1 copy goes this way. Same here, 1 copy that way, 1 this way, 1 that way, 1 this way, 1 that way, 1 this way. And then we end up with, this is a cell and this is a cell, and they divide into 2 new cells, and we end up with 4 daughter cells here. So that's pretty much the process of meiosis. To help us understand it a bit more, we're going to watch an animation that sort of shows this, because it's always helpful to see things moving in these sorts of processes.
Meiosis is a special type of cell division that produces sex cells, also known as gametes. Sex cells are needed for reproduction when an egg cell joins with a sperm cell to form a whole new organism. Most cells in the human body have 46 chromosomes, but sex cells only need half this number. That way, the new baby gets 23 chromosomes from its mother and 23 chromosomes from its father to form a full set. Meiosis involves 2 cycles of cell division, called meiosis I and meiosis II. Each cycle involves the same 4 stages as mitosis, PMAT, with some important differences. Just like mitosis, the parent cell copies its DNA before cell division begins, so each single-stranded chromosome becomes double-stranded. In prophase I, the chromosomes condense and join together in homologous pairs. Each pair contains 1 chromosome from the father and 1 from the mother, with different copies of the same genes. Genes are swapped within each pair in a process called crossing over. The nucleus begins to break down. In metaphase I, the pairs of chromosomes line up in the middle of the cell. Fibers attach the chromosomes to 2 bodies on opposite sides of the cell. In anaphase I, the fibers separate the pairs by pulling them apart. In telophase I, 2 nuclei form, and then the cell splits into 2 cells, each with 23 double-stranded chromosomes. In meiosis II, each cell will divide again to form 4 cells with 23 single-stranded chromosomes. In prophase II, the chromosomes condense and the nucleus breaks down. In metaphase II, the chromosomes line up in the middle of the cell. In anaphase II, fibers pull single-stranded chromosomes apart. And in telophase II, nuclei form, and finally, the cells divide. The 4 sex cells produced by meiosis are genetically different to each other and to the original cell. This genetic variety helps explain why siblings can look and behave so differently from one another.
Alright, so hopefully you have a better understanding of meiosis from that as well, and in fact you have a better understanding of mitosis and meiosis. We're going to pause here, and this will be the end of part 2. There is a part 3, so you need to make sure you come back for that, and we'll go over a few more little details that come along with all this in part 3. So I'll see you there.